Welcome back, sqadians. We are back again with the continuation of our biochemistry series, where we are going to discuss HDL metabolism today. But before discussion of HDL metabolism, let's revise some basic concepts for better understanding. We know that there are three major macronutrients needed to fulfill the demand of energy by our body that includes carbohydrates, fats, and proteins. Our concern is with fats here. Just revise the structure of fats in your mind. 1 glycerol plus 3 fatty acids combine to form triacylglycerol, the ultimate form of fat. Fats can be in the form of triacylglycerol, cholesterol, fat-soluble vitamins, and cholesterol esters. This dietary, exogenous, or endogenous fat is then absorbed by the intestinal wall cells called enterocytes, where they combine with different proteins and form lipoproteins. Proteins that binds with lipids to form lipoproteins are called apolipoproteins. Question arises: why they need protein for transportation? Well, the answer to this question is that small triacylglycerol chains can move directly into liver by hepatic vein, but large fat molecules are extremely hydrophobic, so they can't move freely within the hydrophilic atmosphere of blood. They need some carrier. That carrier here is protein, that together with lipid called lipoproteins. They differ in lipid and protein composition, size, density, and site of origin. They are mainly composed of two portions. Lipids here are mainly triacylglycerol, and cholesterol. Second is apolipoproteins. These proteins are divided into various classes based on their structure and function, like ApoC1, ApoC2, ApoA1, etc. They perform a number of diverse functions, such as providing recognition sites for cell surface receptors and serving as activators or coenzymes for enzymes involved in lipoprotein metabolism. In our previous video of VLDL and LDL metabolism, we have learnt about all lipoproteins. Let's revise them briefly. Large chylomicrons, which are mostly lipid, transport dietary lipids to the liver and other tissues. The liver produces triacylglycerol-rich, very low-density lipoproteins, VLDL. As they circulate in the tissues, VLDL give up their triacylglycerols and, by passing an intermediate stage, becomes cholesterol-rich low-density lipoproteins, LDL, which are taken up by tissues. High-density lipoproteins, HDL, the smallest and densest of the lipoproteins, transport cholesterol from the tissues back to the liver. Here, our focus will be on HDL. The cholesterol-rich LDL particles are notorious as the bad guys of lipoprotein metabolism, as they transport cholesterol from liver to the peripheral tissues. On the other hand, HDL particles enjoy the reputation as the good guys. This is because the function of HDL is to remove surplus cholesterol and transport it to the liver for disposal as bile salts. So, what is HDL? HDL stands for high-density lipoproteins. HDL are the smallest and densest lipoproteins that comprise a heterogeneous family of lipoproteins with a complex metabolism. HDL particles are formed in the blood by the addition of lipid to ApoA1, an apolipoprotein made and secreted by the liver and intestine. ApoA1 accounts for approximately 70% of the apolipoproteins in HDL. As HDL play a very important role in our body, let's move ahead and learn how HDL perform a number of important functions. That includes the following: apolipoprotein supply, non-esterified cholesterol uptake, cholesterol esterification, reverse cholesterol transport. Now we will discuss all these one by one, starting with apolipoprotein supply. HDL particles serve as a circulating reservoir of ApoC2 that is transferred to VLDL and chylomicrons, and ApoE that is required for the receptor-mediated endocytosis of IDL. Next, non-esterified cholesterol uptake. As we know already, that nascent HDL are disc-shaped particles containing primarily phospholipid and ApoA, C, and E. They take up cholesterol from non-hepatic, peripheral tissues and return it to the liver as cholesterol esters. Next is cholesterol esterification. The cholesterol taken up by HDL is immediately esterified by the plasma enzyme lecithin cholesterol acyltransferase, LCAT, also known as PCAT, in which P stands for phosphatidylcholine, the source of the fatty acid. This enzyme is synthesized and secreted by the liver. LCAT binds to nascent HDL and is activated by ApoA1. Let's see how this reaction proceeds. LCAT transfers the fatty acid from carbon-2 of phosphatidylcholine, present in nascent HDL, to cholesterol. This produces a hydrophobic cholesterol ester, which is sequestered in the core of the HDL, and lysophosphatidylcholine, which binds to albumin. Note that this esterification maintains the cholesterol concentration gradient, allowing continued efflux of cholesterol to HDL. Our next important function of HDL is reverse cholesterol transport, RCT. As the name indicates, in reverse of LDL role, that is, transport of cholesterol from liver to peripheral tissues, it reversibly transports cholesterol from peripheral tissues to the liver. The selective transfer of cholesterol from peripheral cells to HDL and from HDL to the liver for bile acid synthesis or disposal through bile is a key component of cholesterol homeostasis. This process of reverse cholesterol transport, RCT, is in part the basis for the inverse relationship seen between plasma HDL concentration and atherosclerosis, that is narrowing and deposition of arteries by plaque buildup, and also this process is responsible for the designation of HDL as the good cholesterol carrier. Interesting thing to note here is that exercise raise HDL levels that has atheroprotective property. The atheroprotective properties of HDL include reverse cholesterol transport, as discussed earlier, reduction of oxidative stress by increasing uptake of lipid peroxides and by inhibiting LDL oxidation, inhibition of vascular inflammation by preventing cholesterol deposition, so there'll be no activation of macrophages and ultimately no release of inflammatory mediators that are cytokines. That's the end of the important functions of HDL. Now let's move forward and learn how HDL scavenges cholesterol. HDL scavenges cholesterol from two sources. Firstly, it will do this through lipoprotein lipase, that is an enzyme. Lipoprotein lipase activity primarily hydrolyzes the triacylglycerol content of lipoproteins to form fatty acids and glycerol. However, in the process it liberates some cholesterol, which is incorporated into HDL particles and is transported to the liver for disposal. Secondly, HDL scavenges cholesterol through ABC transporter proteins. ABC transporter proteins are a family of proteins characterized by an ATP-binding cassette. The bound ATP is hydrolyzed in a process coupled to transport of their substrate. ABC proteins use energy from ATP hydrolysis to transport materials, including lipids, in and out of cells and across intracellular compartments. One such protein is the cholesterol transporter known as ABCA1. It is found in many tissues where its function is to transfer excess cholesterol to HDL particles. Then, HDL particles proceed to the liver for disposal. Now let's move ahead and learn the metabolism of HDL. HDL is the smallest, densest lipid transport vehicle that are assembled in the liver and intestine from apoproteins A1, C2, E and phospholipids. HDL donate apoproteins to chylomicrons and VLDL particles. HDL also play a very important role in cholesterol metabolism by transporting excess cellular cholesterol through the bloodstream to the liver for excretion in bile. Now, let's see how cholesterol modified itself for transport through HDL. We know that cholesterol undergoes esterification prior to enter liver. Here question arises, why, cholesterol need esterification. The polar hydroxyl group at C3 is hydrophilic, and cholesterol tends to locate itself superficially in lipid phases, such as the phospholipid monolayer. However, molecular shielding of this hydroxyl group, by covering it with fatty acid by an esterification reaction, allows cholesterol to be packaged in the hydrophobic interior of lipoproteins, allowing for increased transport capacity. The reaction product of this reaction is a cholesterol ester. This occurs intracellularly, and it is catalyzed by acyl coenzyme A, cholesterol acyltransferase, ACAT. Until now, we have cleared our concepts on the cellular cholesterol clearance, that is how cholesterol is packaged inside the HDL and then transported towards liver. Now we will move forward to know about how it will be removed from the body. When it enters in the liver in the form of cholesterol esters, cholesterol is metabolized to form bile salts, which are excreted in the bile duct that enters small intestine. The bile salts emulsify fats in the intestine, which renders them available for hydrolysis by pancreatic lipase, which is secreted into the gut. About 95% of the bile salts are absorbed into the hepatic portal vein, a vessel that moves blood from the spleen and gastrointestinal tract to the liver, and are recycled to the liver by the enterohepatic circulation. About 5% of the bile salts are lost in the feces. The enterohepatic circulation can be interrupted by anticholesterol agents. These are positively charged resins that bind to the negatively charged bile salts and form complexes. The resin bile salt complex is then digested in the feces. Now it officially brings us to the end of our topic, where we have discussed very important concepts today. We have learnt all the important functions of HDL, are two main sources through which HDL scavenges cholesterol, HDL metabolism, in which we go through the cholesterol modification for transport, then its process of elimination, and lastly, disposal of cholesterol as bile salts. This is the full jam-packed knowledge video, just for you guys. Stay tuned for more informative videos like these, only on sqadia.com.